Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
The Wobble Hypothesis explained. Table of Contents. Introduction to the Wobble Hypothesis. The Wobble Hypothesis statement. Wobble base pairs. Significance of the Wobble Hypothesis. Introduction to the Wobble Hypothesis. There is more than one codon that codes for a single amino acid, a feature known as the degeneracy of the genetic code. To explain the possible reason for this degeneracy, Francis Crick proposed the Wobble Hypothesis in 1966. According to the Wobble Hypothesis, only the first two bases of the codon form precise and specific pairings with the corresponding bases of the anticodon on tRNA. The third base of the codon, however, does not always follow strict base pairing rules. Instead, it may wobble, meaning it can move or sway unsteadily, allowing flexible pairing. This wobbling at the third position enables a single tRNA molecule to recognize and bind with more than one codon. As a result of this flexibility, although there are 61 codons that specify amino acids, the number of tRNA molecules required is much fewer, approximately 40 tRNAs, because of the wobble phenomenon. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. The Wobble Hypothesis statement. The Wobble Hypothesis states that the base at the 5' end of the anticodon is not as spatially restricted as the other two bases, allowing it to form hydrogen bonds with several possible bases located at the 3' end of a codon. This flexibility leads to several important conclusions about codon-anticodon pairing. The first two bases of the codon form normal, canonical, hydrogen bonds with the second and third bases of the anticodon. At the third codon position, less stringent pairing rules apply, allowing non-canonical base pairing to occur. The Wobble Hypothesis therefore introduces a more flexible set of base pairing rules specifically at the third position of the codon. This relaxed base pairing, or wobble, enables the anticodon of a single tRNA to recognize and pair with more than one mRNA codon. The base pairing rules for wobble position are as follows. First base U on the anticodon can recognize A or G on the codon. First base G on the anticodon can recognize U or C on the codon. First base I, inosine, on the anticodon can recognize U, C, or A on the codon. Crick's hypothesis, hence, predicts that the initial two ribonucleotides of triplet codes are often more critical than the third member in attracting the correct tRNA. Wobble base pair. A wobble base pair refers to a pairing between two nucleotides in RNA molecules that does not conform to the traditional Watson-Crick base pairing rules. The four main types of wobble base pairs are guanine uracil, hypoxanthine uracil, hypoxanthine adenine, hypoxanthine cytosine. To maintain nucleic acid nomenclature consistency, the letter I is used to represent hypoxanthine, as hypoxanthine serves as the nucleobase of inosine. Inosine exhibits the true wobble property because when it is the first nucleotide in the anticodon, it can pair with any of three different bases, U, A, G, C, or A, in the codon, allowing greater flexibility in codon recognition. Download our microbiology study app made for researchers like you. Significance of the Wobble Hypothesis. The Wobble hypothesis is significant because it allows broad specificity in codon recognition, compensating for the limited number of tRNAs available in the body. Wobble base pairs play essential roles in various biological functions, which have been clearly demonstrated in Bacterium Escherichia coli. The thermodynamic stability of a wobble base pair is comparable to that of a normal Watson-Crick base pair, ensuring structural reliability during translation. These wobble interactions are fundamental to RNA secondary structure formation and are crucial for accurate and efficient translation of the genetic code. Wobbling facilitates faster dissociation of tRNA from mRNA, thereby enhancing the rate of protein synthesis. The existence of wobble also reduces the potential impact of codon misreading. For instance, if the leucine codon, CUU, were misread as CUC, CUA, or CUG during mRNA transcription, it would still be correctly translated as leucine during protein synthesis, minimizing errors in the resulting protein. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!